Hello everyone and welcome to Lab 1, the beginning. The purpose of this lab is to introduce the major divisions of the central nervous system, the forebrain, midbrain, hindbrain, and spinal cord. We also want to identify key landmark structures within each of those divisions and understand the development and structure of the brain's ventricular system. The central nervous system, composed of the brain and spinal cord, is derived from a specialized embryonic structure called the neural tube, which originates from neuroectoderm. By understanding the basic layout of the neural tube, we can establish a framework for conceptualizing the complex 3D structure of the adult CNS. The neural tube can be divided into two main regions, a rostral portion and a caudal portion. The caudal portion of the tube will eventually become the spinal cord, which is the section of central nervous system found within the vertebral column. The most caudal point of the neural tube will end up being the conus medullaris of the spinal cord, seen here. We will be taking a more detailed look at the spinal cord in Lab 3. The rostral end of the neural tube gives rise to the brain, defined as the portion of CNS contained within the skull. Early on, the rostral end of the neural tube is formed by three vesicles, the prosencephalon, the mesencephalon, and the rhombencephalon, which give rise to the forebrain, the midbrain, and the hindbrain, respectively. The rhombencephalon, or hindbrain, gives rise to three major structures, which can be seen here on a mid-sagittal section through the adult brain. The first is the medulla, which is the most caudal portion of the brain. It connects to the spinal cord here at the spino-medullary junction. Next we have the pons, which can be distinguished by this large ventral bulge called the basis pontis. Finally, the cerebellum, or mini-brain, which extends off the pons dorsally, expands to cover both the pons and the medulla. The mesencephalon, which simply develops into the midbrain, can be seen here, rostral to the pons. The prosencephalon is where things get tricky, because there's an intermediate step. During development, the prosencephalon differentiates into two daughter vesicles that each contribute to the adult forebrain. These are called the diencephalon and the telencephalon. The diencephalon, which abuts the midbrain, eventually gives rise to the hypothalamus and the thalamus. If we look more closely at the diencephalon on mid-sagittal section, we can see the thalamus just rostral to the midbrain. The hypothalamus will then be inferior and slightly anterior to the thalamus. In addition to the thalamus and the hypothalamus, the diencephalon also develops bilateral outpouchings called the optic vesicles. These vesicles eventually become the retina and the optic nerves. If we look at the ventrum of the brain, the optic nerves can be seen here. If we trace the optic nerves back towards the diencephalon, we run into a midline structure called the optic chiasm. The optic chiasm is formed from fibers that originate in the nasal retina and cross the midline to reach the contralateral side of the brain. It is important to note that the retina is the only part of the central nervous system that can be visualized from the outside world, and therefore can be used clinically as a window into the CNS. The remaining structures of the forebrain, highlighted here, are derived from the telencephalon. The telencephalon begins as a bulge off the rostral point of the diencephalon, which then collapses in the middle to form two hemispheres. These hemispheres then expand to form the large cerebral hemispheres of the brain. To get a better understanding of how the cerebral hemispheres take shape, we can take a cross-section of our primitive neural tube model. Here you can see the diencephalon along with the two telencephalic hemispheres bulging out like Mickey Mouse ears. As the telencephalon expands, the upper portion of the telencephalon will form the gray and white matter of the cerebral cortex. The gray matter, containing neuronal cell bodies, is organized into an undulating outer rind that forms the exterior surface of the forebrain. The white matter below is composed of myelinated axons that connect the cortex and lower structures. The lower portion of the telencephalic hemispheres will develop into the amygdala and several deep structures called the striatum and globus pallidus that are part of the basal ganglia. It is important to note that as the telencephalon expands, the space deep to the cortex is filled with axons that connect to distant structures. A group of axons originating in the frontal, parietal, and occipital lobes eventually crosses the midline to form interconnections between the two hemispheres, thus allowing them to communicate. These axons form a massive band called the corpus callosum. Because the corpus callosum crosses the midline, it is called a commissure, or a commissural structure. On gross specimen, we can visualize the corpus callosum as it bridges the two hemispheres by gently pulling them apart. We can also see it here on a mid-sagittal section, rostral to the thalamus and just deep to the cortex. Another large white matter tract develops bilaterally to connect the telencephalic hemispheres to more caudal structures. Beginning from neurons in the cerebral cortex, these axons travel deep into the cortex in a fan-like shape, forming a structure called the corona radiata. As these axons continue caudally, they then pierce the striatum, splitting it into two parts, the caudate and the putamen. This portion of the tract is called the internal capsule. The axons of the internal capsule then proceed ventrally and caudally into the cerebral peduncles that form the base of the midbrain. We will take a more detailed look at the cerebral peduncles in Lab 2. Another important telencephalic structure is the amygdala, which can be seen here below the basal ganglia within the temporal lobes of the cortex. Now that we've had an overview of the basic structures of the brain, let's take a look at the space inside the brain. Remember, within the neural tube there exists a fluid-filled space. As the brain develops, the fluid-filled space persists and takes shape along with its neighboring structures. Thus, in the adult brain, each of the divisions we have just seen has a corresponding fluid-filled space, or ventricle. The fluid that fills the ventricles is called cerebrospinal fluid, or CSF. CSF is produced by a structure called the choroid plexus, which can be found in all of the ventricles except the cerebral aqueduct. The choroid plexus is composed of a complex network of capillaries surrounded by a specialized choroid epithelium. The choroid epithelium filters the blood, allowing only specific components within the blood to pass through into the ventricular space. This process not only produces CSF, but helps form the blood-CSF barrier. Due to a pressure gradient established within the brain, CSF tends to flow rostrally to caudally, beginning with the telencephalon. The telencephalon houses a pair of lateral ventricles, one in each hemisphere. They can be seen here first on coronal cut, and now on a horizontal cut. The reason why the telencephalon has two paired ventricles can be seen with our original diagram of telencephalic expansion. As we saw before, the telencephalon develops from an initial bulge off the diencephalon that then collapses into two separate hemispheres. Though the center collapses, each hemisphere maintains a lumen that is continuous with the lumen of the diencephalon, which will become the third ventricle. The openings through which the lateral ventricles of the telencephalon and the third ventricle of the diencephalon communicate are called the foramina of Monro. Though they may look simple enough on coronal section, the lateral ventricles actually have a very complex 3D shape as a result of the rostral and caudal expansion of the telencephalic hemispheres. Looking at the neural tube from the side, we can watch the telencephalon expand anteriorly and inferiorly, as well as posteriorly. Once the hemispheres have expanded posteriorly, the telencephalon undergoes a process called temporalization, where the hemispheres curve inferiorly and then back anteriorly, forming the temporal lobes. You can picture it kind of like a ram's horn. Due to this temporalization, the lateral ventricles within each hemisphere are stretched into a sort of C shape, very similar to the University of Chicago C. The caudate and putamen, which we saw earlier, are also stretched in a similar manner and follow the lateral ventricles closely, beginning in the front of the telencephalon and ending in the temporal lobe. Another important result of temporalization is the creation of the velum interpositum. The velum interpositum is the space created between the expanding telencephalon and the underlying diencephalon. This space is technically outside the brain and houses a variety of blood vessels. From the lateral ventricle, CSF flows through the foramina of Monro and into the diencephalic third ventricle. The third ventricle lies in the midline of the diencephalon and is walled in on either side by the thalamus. This can be seen here on cross-section with the third ventricle sandwiched between the two lobes of the thalamus. On mid-sagittal section, we've actually cut through the third ventricle because it lies in the midline. What you're seeing here is one of the lobes of the thalamus forming the lateral wall of the third ventricle. From the third ventricle, CSF flows to the mesencephalon, or midbrain. Unlike the ventricles we saw in the telencephalon and the diencephalon of the forebrain, the space within the mesencephalon does not expand and remains a narrow channel. This is called the cerebral aqueduct, and can be seen here on mid-sagittal section. Remember, the cerebral aqueduct of the midbrain is the only ventricle without choroid plexus. The cerebral aqueduct of the midbrain connects to the ventricular space of the rhombencephalon, or fourth ventricle. Looking at the rhombencephalon in cross-section, the ventral portion will become the medulla caudally and the pons rostrally. Progenitor cells in the rhombic lip on either side of the rhombencephalic lumen will proliferate to form the cerebellum. The cerebellum contains an enormous number of cells, probably more than half of all the cells in the CNS. The cerebellum is correspondingly enlarged and comes to overhang all of the pons and part of the medulla. It remains attached to the pons at 2 points called the cerebellar peduncles. Thus, the fourth ventricle of the adult brain is bounded by the cerebellum dorsally, the pons and medulla ventrally, and the cerebellar peduncles laterally. If we remove the cerebellum from the dorsum of the hindbrain here, we will be looking onto the floor of the fourth ventricle. Notice that the ventricle has a rhomboid shape, and it is therefore referred to as the rhomboid fossa. At the base of the fourth ventricle, CSF exits the ventricular system through 3 foramina: the foramen of Magendie, located caudally in the midline, and the 2 laterally located foramina of Luschka. Because the lumen of the spinal cord, called the central canal, becomes occluded during development, CSF is forced to exit through the foramina of the fourth ventricle into the subarachnoid space. Once in the subarachnoid space, the CSF bathes the convexity of the brain and the spinal cord, until it is reabsorbed into the circulation through arachnoid granulations that project into the dural sinuses. This completes the material for Lab 1. Feel free to use this video as well as the image library to help complete the exercises and navigate your dissection.